All right, recording begins. It is September 15th. This is Biology 105. Thank you all for making it once again. All right, so we are going to try to wrap up our biological macromolecules talk today. That'll be the week 3 material. Get into the week 4 material, which is all cell stuff. All right, it's going to be great. You're going to love it. Carbohydrate graphing assignments. First real, like had to submit something thing. You got another lab this week. This has a protein graphing assignment, okay? In this week's lab, however, it's slightly different in that you're going to have to graph, okay, and analyze some data in order to answer questions, okay? But you're not uploading the figure. Just a little different. I'll give you another refresher on Wednesday, but don't forget, you have these lab assignments. All right, these lab assignments are there. This week, what's going on in our module? We've got laboratory materials linked right there. Do not forget to watch the pre-lab video. This week's lab is going to be about protein quantitation and breaking open cells with osmotic pressure. It's going to be interesting. We'll have talked about, bless you, we'll have spoken about amino acids and proteins by the time this lab hits, so you'll understand a little bit of what we're doing. Once again, I got a standard curve and analysis tutorial video showing you exactly how to do the graphing and the analysis part, all right? It's linked here, it's linked in the assignment, and it's linked in the question where you need the video, okay? So it's everywhere, this tutorial video. Watch the tutorial video, all right? It'll help you get through the steps that you need. Once you start this assignment, there's a time limit, okay? So you want to always be aware of that. You can't pause the assignments, okay? That would defeat the purpose of a time limit on the assignment. There's no pausing the assignment, so start it when you're ready to sit down and do the analysis. But hey, you could watch the video beforehand and even analyze your data beforehand. So when the question hits, you're ready to go. Just saying. Any questions about the lab for this week? Print the protocol, watch the pre-lab video, do the stuff that you've done every single week, and we'll just keep on crushing it. Sound good? I'm super into it. I might have overcorrected. I might be being too intense today because I was so low energy on Friday. But, hopefully it doesn't come off as too aggressive. All right? Also, reading quiz for Chapter 6. Sound good? Any questions before we get into the material? Yes? I'll get you next.
More or less similar in format to the carbohydrate one?
It is not. I will show you a preview of the assignment. So it's got general multiple choice questions, okay, all based on the protocol. And then it tells you, take this data file, analyze it, and then use it to answer these questions. Less subjective grading, and it'll grade immediately, which is always nice. I got one ahead of you, yes.
So is that also 2 attempts, like the other kind of quiz stuff? Or?
2 attempts.
And also my second question is, for the exam Monday, will it be on week 1 through 3 material, or?
Everything on the study guide.
Okay.
Everything on the study guide. Always the answer to what's on the exam is everything on the study guide. Solid. Of course, yes.
With this lab, last time, the last week, the lab took a very long time. Like some groups, on the 3-hour period. Is it going to take a similar amount of length, or be shorter, or if for some reason, if it is really long, will we have some information to go off of if we don't finish in time?
Yes. Okay. So the answer to your question is, expect the labs to always take the full 3 hours this semester. That's the first thing. They'll go faster if everybody on your bench has watched the video and if you plan out your activities. So when you look at your protocol, and if you look through the whole protocol and see, which you should be doing before the lab, and you see, oh, there's a 30-minute incubation step, and we have 2 tasks we have to do. Well, we could do this other task during the 30-minute incubation step. What do you think about that, benchmates? I think that's a great idea. We are collaborating now to be more efficient. You see what I'm saying? So some of this stuff is going to be on your end. If you don't finish, everything you need to complete the lab assignment is in the lab assignment. The protocol has all the information that you need to know from the lab. The pre-lab video has all the information you need to know from the lab. So all the information you need to be prepared for the lab midterm and the lab final and to complete the assignment is already available to you. Cool?
Yeah, it's just our lab group had 3 people instead of like the 5, so we weren't really able to finish it, even though we were all working overdrive.
Totally. And yeah, I'm not trying to say like these labs, they tend to be time consuming. We met last week and talked about last week's lab, and we think we've made some changes that'll make it go more quickly next semester, but that does not help you this semester at all. This one, we talked about it in the TA meeting, and it should be done by the time the lab ends. We haven't made a bunch of changes, and this one usually runs pretty effectively. There is a 30-minute incubation step, though, so it's something to think about. Cool. Additional questions? All right. There is an exam. I'm so happy you mentioned that. Thank you so much for bringing that up. Exam 1 is a week from today. All right. There was an announcement made by your SI instructor about a review session that you can sign up for if you want to go to the review session. The SI instructor has a copy of the exam. They know what's going to be on the exam, but so do you, because it's in the study guide, which is posted in the week 5 module right here. Cool? Question, and I'll get you next. Yeah. Multiple choice, 25 questions. Yeah. Sorry to cut you off. Huh? Scantron 882E form. You sure do. You should bring a Scantron 882E form. It's the hot dog one. It's green. It's a green hot dog. By which I mean it's the one of the narrow forms. That's it. You want to hold that up for everybody? Yep. Those of you with forms, hold those up in the air. Proudly wave them about. Yes, woo. We're going wild in this classroom today. I know. Thank you so much. Saul, did that answer your question? Once again, I apologize for how intense I'm being today. I got so much sleep. I feel great. All right, I got a few questions, but I'll be there for you. But I see 2 in the back, side by side. Please answer your questions or ask them at the same time. Synchronize your voices. I am not sure. All I know is what's in the announcement. I would say check out that Google sheet. How about you?
I don't have a question. I just thought it was a good public information that the Scantrons are sold above the depot, which I didn't know for all of last year.
Oh, yeah. Thank you for mentioning it. They're above the depot. That's one source. I believe the Rose House and the Learning Center also have them, okay? I'm trying to keep track of whose hands are up first. I got a hand here, and then I'm going to go over here, and then I'm going over here, then I'm going far to the back corner. Yes?
Is the exam similar in structure to the reading quizzes, like similar structure?
Yes, but I would say easier because you don't have a book to reference. It's all in your head. So I've tried to select questions that are easier. Yeah, I had one here, right? And then I'm going to go boom, 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 boom. Okay? Cool, yeah.
I don't know if you said, but they're also free in the Rose House. You can get them in like bulk.
Rose House.
4 or 8 at a time.
Yeah, Rose House, free Scantrons. If you have accommodations, the testing center, the CDRC center, is usually cool about giving you Scantrons too. There are sources for free Scantrons. Absolutely, 100%. Or you can pay for them above the depot. I had one here, and then I'm going to the far back, then I'm coming down. That's my plan. Yes? Oh, they're in the Rose House. Yeah, solid. Awesome. Thank you very much. Yes?
Another place is the Social Justice and Equity Center by the library. They have free Scantrons in the computer.
That's the one on the corner, right? Like on the corner if you're going up this way on that road?
Yes.
Awesome. Social Justice and Equity Center, another source for Scantrons. I'm so happy they're everywhere and that people are using Scantrons again, because Scantrons are great. I love them so much. Yeah, I just love Scantrons. Yes, question.
I assume not, because it's not on the study guide. And so there's not going to be chemistry and math on the exam.
It's not going to be chemistry math on the exam. If it's not on the study guide, it's not on the exam. There you go. Yeah, wait, you had a question like a... Oh. Oh, awesome. I'm happy to hear that. Cool. how are we doing, folks? Additional questions? I know it's the first exam. It's big. It's scary. It's points, you know, they're all out there. It's multiple choice. It's a Scantron exam. Some of you have never used a Scantron before, and that's okay, you know? Oh, yeah. Once per semester, if you fail an exam, you can do an extra credit report on the questions you missed to get half of the points back that you missed. You got to get 70% or below. Don't worry about the details of that because none of you are going to get under 70%. And if you do, you come talk to me after that happens. You talk to me one-on-one to get the details. Okay? Cool? All right, very good. Questions, no? All right. Anything else? We got to get into our lecture, you know? Yeah, I'll just look over the PowerPoint slides. It's fine. Cool. Everybody good? All right, I'm very excited to get into this. We have to get through some serious material. We got to get into phagocytosis. Oh, yeah, we're going to be fine. We're definitely going to get through there. No problem. Okay. Biological macromolecules. It's so exciting. We've been talking about, we have not gotten to proteins. We have been talking about carbohydrates and lipids. That's what we've been talking about. All right, remember the main thing about the lipids that's going to be super relevant is that they are hydrocarbons and they are hydrophobic. All right, because they're made up mainly of these carbon and hydrogen linked tails or fatty acids. All right, cool. Okay, we need to get into saturated and unsaturated fats, what they are, why they have different health implications, and how they behave differently when when a bunch of them group up. All right, let's go. So I ended with this. The building block of the fatty acid, of the lipids is a fatty acid. But the fatty acids don't fit into that polymer-monomer framework because remember, with the monomers, they're joined in a big chain, all right? And each monomer can be different, and that gives the polymer special emergent properties. But with the lipids, we have these fatty acid chains. They're not joined in a sequence like that, all right? So the fatty acid is like the building block of the lipids, but it is not a monomer because it's not joined into a sequence in a polymer. Does that make a little bit of sense? Here's what an actual fat looks like a triglyceride. It's multiple fatty acids all joined to a glycerol molecule. All right? Awesome. Okay. So, the fatty acids. There are saturated fatty acids and there are unsaturated fatty acids. And here's the way to think about it. A saturated fatty acid is saturated with hydrogen. It has the maximum number of hydrogen atoms that it could have. There are no double bonds in a saturated fatty acid, except for the one over here in the carboxyl group, but let's just focus on the hydrocarbon portion, okay? Cool. So fatty acids can be different lengths, and they also differ in whether or not there are double carbon-to-carbon bonds, okay? Because think about this, right? If there's a double bond between this carbon and this carbon right here, well, then there won't be a hydrogen here or here. So you would not have the maximum number of hydrogen atoms. I'll show you an example of that as well. So saturated fatty acids have no double bonds. Unsaturated fatty acids have double bonds. And remember our cis-trans isomers, right? They can exist in the cis or the trans form. And remember when I did my thing with my hands and I put them up and I was like, look, a double bond you can't rotate around. So that makes this molecule here different from this molecule right here, right? So in a cis unsaturated fatty acid, cis meaning same, the orientation on either side of the double bond is the same, all right? You end up with a bend, okay, or a kink in the fatty acid. In a trans unsaturated fatty acid, all right, meaning opposite orientation on either side of the double bond, okay? The fatty acid chain still lies flat in a space-filling model. So it's the difference between this straight, right? A saturated and a trans-unsaturated fatty acid will both lie flat and not take up as much space as a cis-unsaturated fatty acid, which has a bend in it, all right? The analogy I always use is branches, like a tree branch that has a bunch of like bends in it, okay, versus a dowel, like a straight rod, you know? If you wanted to pack a bunch of dowels into a bucket, you could fit way more dowels in the bucket than you could branches. Does that sort of make sense? So saturated fatty acids and trans-unsaturated fatty acids, and therefore saturated fats and trans-unsaturated fats, can be packed at a higher density. They're more stable because they don't have all of these bends that are like interfering with one another and preventing tight packing. All right, bless you. So the saturated and the trans-unsaturated can be tightly packed, reach higher densities, they're more stable, okay? They form solids at higher temperatures because they're more stable. So saturated and trans fats are like, saturated fat's like a butter. It's solid at room temperature because the molecules can pack more tightly more easily. A cis unsaturated fat would be like an olive oil, okay? And in that olive oil, the molecules can't pack as tightly so it stays liquid. Cool? Very good. Very, very good. So now, looking at an actual fat, it's 3 fatty acids attached to a glycerol. Now, each one of these fatty acids can be a different length, and it can differ in whether it's saturated or unsaturated. All right? So in a saturated fat, they're all saturated fatty acids. In an unsaturated fat, one or more of those fatty acids has one or more double bonds. And those double bonds can result in a cis or a trans isomer of the fatty acid. Yes?
That's what I was gonna ask. Or I was gonna ask if cis or trans like was linked to saturated or unsaturated because you said
Yes.
okay.
Oh, sorry, I don't mean to cut you off. No, please, please.
Just like you said, trans was butter and cis was olive oil, and I was like, wait, but now it's saturated and unsaturated.
So what I meant was that what I meant to say, I misspeak sometimes for sure. Saturated fats are like butter, okay? But a trans unsaturated fat will also behave like a saturated fat. So it will also be solid at room temperature usually. So like an I can't believe it's not butter could be what was an unsaturated fat that they made into a saturated fat. So now it'll be stable at room temperature, and that's better for some reasons. All right, solid. So the unsaturated fats, right? Oh, just one second. Oh, sorry, just one second. The unsaturated fats can be cis or trans, but a saturated fat cannot be cis or trans, all right? Cis and trans fats are specifically unsaturated fats because they relate to the arrangement around a double bond, and a saturated fat does not have any double bonds. That was the last piece of the answer to your question that I wanted to give you. Please, yeah. Oh, did I? Like, no big deal. I can just tell, you know? Just, yeah. All right, solid. Saturated, unsaturated fats. It's tricky, but it's super important. That's why I dwell on it, okay? It has repercussions for, personal health. This is the underlying reason that, well, there's another reason, right? But this is one of the reasons that we say unsaturated fats, olive oil is healthier for you. Use it instead of butter, right? Because this more liquid, less stable fat, all right, can be processed and moved through your body a little easier. It's more liquid, it's more fluid, all right? As opposed to the saturated fats and the trans fats, which can form solid plaques inside of your body. Because guess what, folks? Your blood is an aqueous solution. So these molecules are already insoluble in that aqueous solution. So if they can pack tightly and become super stable, like in a saturated or a trans fat, well, then they can glob together, get stuck in your bloodstream, get stuck in your body, form plaques, no good. Right? Cool. Hoo-wee. Let's keep on going. Which of the following two fats is the cis fatty acid? All right? Remember, it's all about the arrangement around the double bond. So A or B? That is correct. A. Very good. Trans fats. Oh my God, cholesterol. Is that a vine that people still enjoy? Okay, good. Oh my God, trans fats. It's fun. We have fun here. Okay? Trans fats, they still put it on labels of food like, hey, we got no trans fats in here, even though it's less and less common to utilize these things because of the recognition of the negative health impacts, the increased rates of regulation. You don't want to change your recipe when you sell in different countries if you can avoid it, right? You want to make one Cheeto everywhere the same way. Have it always be the same Cheeto. Anyways, trans fats. They're fascinating. They're fascinating. Okay? The person who developed the process for creating trans fats, I believe, might have won a Nobel Prize, because what this did is it let you take unstable, unsaturated, like vegetable oils and convert them into a solid that could then more easily be shipped. So you could get these high energy fats through the country redistributed. It's pretty interesting, okay? But trans fats, they behave like saturated fats, but they also have double bonds. They're more stable. They're worse for you. So it's better to just use butter anyways if you're going to use a saturated fat. Now, there's all sorts of interesting processes where you can bubble, essentially you bubble hydrogen through through a unsaturated, cis unsaturated fat, right? That's like liquid. You bubble, usually it's nickel hydride or some compound like that. Then you have to deal with the extra nickel inside it. It's weird. But you can convert an unsaturated fat with trans bonds, okay, or cis bonds, into a saturated fat and occasionally a trans fat. Here's the idea. Here's the idea. I've got a big old vat. Let's go to the theater of the mind again. Here we go, we've teleported. I've got a big old witch's cauldron right here. And underneath it, I am bubbling this hydrogen-containing gas. And it's a liquid already. It's this liquidy vegetable oil. And I'm going, and I'm churning it, and I'm churning it. And then, occasionally, some of those fats get converted into saturated fats, but also they get converted into trans fats, and they solidify. And then I just go, and I scoop up these big blocks of now solid fats. And I go, it's sourced from a vegetable, so it's better for you. Eat it. Put it on your toast. Don't you feel healthy? It's a vegetable-sourced saturated fat. It's lost all of the health benefits of being an unsaturated fat, but it came from a vegetable. So it's good for you. All right? Isn't that great? It's brilliant marketing, and it's all because of an evil cackling witch. William Norman. Wilhelm Norman, I mean. All right, cool. So anyway, trans fats are interesting. Here's a comparison of a cis unsaturated fatty acid with a trans unsaturated fatty acid. All right, and look, they've got different melting points. So oleic acid can't pack as tightly, so it becomes a liquid at a lower temperature. So in general, the cis unsaturated fatty acids or the cis unsaturated fats are liquid at room temperature, and the saturated fats and the trans fats are solid at room temperature. How do we feel about this? Can I answer any questions about it? Yes, please.
I just had a question about so if you boil unsaturated cis fats over hydrogen, like hydrogen oil, what you were doing, so all that.
Yeah.
So that then turns it into either saturated or trans unsaturated by adding hydrogens.
It disrupts the bonds,
Oh, so
but
they can form different, more stable
You got it. And here, yeah.
Goes and also just adds more hydrogen by making more single bonds.
Yes. So the goal is to make it saturated. But when you pull the cube out, you can't tell if it's saturated or trans fats because they behave the same. So you pull them out and you're like, cool, they became solid, we're good, just mix them all together. So you get both.
So it just disrupts the configuration and it configures to a more stable combination.
Essentially, yeah. Yeah. You have to heat it up to destabilize the molecule, and then you pump a bunch of hydrogen in there so that it's more likely that it forms those saturated, that becomes saturated. Awesome. Very good. Yes, and then I'll get you next. Yeah.
Question. So can we go back to the slide with like the trans fat and the unsaturated fat?
Wait.
Aside from the
These are both unsaturated fats, just to be clear, because there's a double bond in both of them.
I was about to say, like, the trans fat, it's literally the exact same molecule, it's just not bent. Is that, like, true?
Yep, these are different isomers. Remember, the cis and the trans are isomers, so they have the same number of each atom. Yeah, we have to think, like, it's all connected, but it's so hard to keep it all connected in our heads.
And the second thing, what about what about Glee clarifying butter? What does that do to it?
That's a great question, and I just don't know. I don't know what the process for making clarified butter is, but I definitely need to look into that.
Yeah, because I heard that behaves much more like an unsaturated fat or oil, and it's used in that manner than normal butter.
Yeah, that makes sense. Doesn't it use like a bunch of centrifugation? Does anybody know about ghee generation? I'll look into it because I'm super curious. I got one ahead of you. Yeah.
Unsaturated trans fats behave like saturated fats.
Yes, in that they are solid at room temperature. Yeah. But they're also worse for you because they're more stable. Your body can't break them down as easily. That's the way to sort of, there's more to it. There's always more to it, of course. But yeah. Got it?
Yeah, do they
Awesome.
behave chemically the same or just physically?
I'm just going to say physically.
Okay.
They behave physically in a very similar way in when they become solid and forming plaques and those sorts of things. Chemically, they behave a little differently in how they get broken down.
Okay.
Yeah. And there are also certain unsaturated fatty acids that are essential for our bodies to process, but that we don't make. And so we have to eat them like vitamins. So when people are like, take your fish oil, you need those omega fatty acids, that's what they're talking about. It's really wild that your body can't just make all the fats you need. Yeah. Question, please.
I think my question was kind of answered partly. So the trans,
Okay.
like the lactic acid, that's solid at room temperature, and then the lactic acid's liquid at room temperature.
Solid at room temperature, liquid at room temperature. Exactly. It's all about how tightly they can pack. Cool. Yes.
Are there any naturally occurring trans fats?
Yes, there are. I don't have a good example for you, but yes, there are. Yeah. Question, please.
Can there be, so with these bonds, it's showing only like one double bond, but can there be multiple double bonds?
Awesome. That's solid. Great. Points for you. Yes, 100%. So that's like the real additional complicating factor here is all of these chains, they can be different lengths. They can be saturated or unsaturated. And then if they're unsaturated, they can have one or more double bonds. So there's all this complication here. And then some of them exist in different, like in an equilibrium. It's biochem stuff. I don't know. I don't fully understand it. I'll be 100% honest with you. But yes, there's all that variability here. Yeah.
Can there be a cis and a trans bond in one?
In one molecule, yes, 100%. Yeah. So there's like a whole numbering scheme where you're like, oh, this is cis 1 trans 6 alpha fatty acid blah blah. You know, once you get into OChem and you're naming the things and you have all the numbering and then the different functional groups indicated, it's very frustrating. Yeah. But so the main difference between the fatty acids, though, is are they saturated or unsaturated, and how many double bonds do they have? Are they cis and trans, right? Solid. Okay. Oh, yeah. Oh, never mind. Look at this. I forgot. I thought I eliminated this slide because this is interesting, but you won't be tested on this or anything. It's what I just mentioned to you in your question, right? But it's also a great indicator of look at the different degree of saturation, of unsaturation in these things. So there are these omega fatty acids. They tell you there's a whole directionality to these fatty acid molecules where there's an omega end and an alpha end, and you number like where are the double bonds. So omega-3 and omega-6 fatty acids, which you'll see as, someone's got fish in it, they'll be like, hey, your cat will get a bunch of omega-3s and omega-6s. And you're like, cool, my cat's gonna love that, all right? They're important, they're like essential, and we don't really make them, okay? Or at least we don't make them in the abundance that our bodies need them. So deficiencies in these fatty acids have been linked to all sorts of disorders. It's just weird to think about a fat as a vitamin, you know? Anyway, omega-6 fatty acid, omega-3 fatty acids. So eat the oils that have the unsaturated fats that are good for you, I guess. That's my take-home message for you. Question, please.
Fish oils wouldn't be considered a natural.
They would be, yeah.
Natural trans fat.
They have, I believe that they have both cis and trans unsaturated bonds in them. Yeah, so that is the answer to your, like, what are there examples of naturally occurring unsaturated fatty acids? Right here, linoleic acid. I should have just said that, That's what we were all thinking, right? Cool. Phospholipids, all right? This is a super important concept. It'll come up again when we do our cell talk. It's so dang important. Check it out. Remember the triglycerides? The triglycerides, I'm going to do it over here so that it's on the video. I'm going to stop pointing at that one. I don't know why it's so attractive to me. Glycerol molecule, okay? You get 3 fatty acids attached to it. That makes a triglyceride. In a phospholipid, you've got the glycerol molecule, but there's only 2 fatty acids attached to it, okay? Where the 3rd fatty acid would be attached is a phosphate and a choline group. And this little group up here, look at that. It's got charge to it. It's polar. It's highly polar. So a phospholipid is what we call an amphipathic molecule. Amphipathic molecule, okay? Meaning it has polar regions and it has nonpolar regions, meaning it has hydrophobic regions and hydrophilic regions. The polar region is hydrophilic, it loves water. The nonpolar region is hydrophobic, it hates water, all right? Here's a phospholipid symbol. This is how I'll draw them, sort of, when I have to draw them on the board. Like a little circle with 2 squiggly legs coming off of it, okay? Indicating the hydrophilic head and the hydrophobic tails. This is going to be so important. It makes up all your membranes, yeah.
Is it always a saturated and an unsaturated fatty acid, or can it be any combination?
Great question. Points for you as well, all right? So it can differ, okay? You can differ in the exact same way of how many, whether it's saturated and unsaturated, how many double bonds there are, and then which ones are cis versus which ones are trans double bonds, right? So there's a ton of variability here, and that will be super important because when these get into a solution and they all group up, if they've got the bends in their fatty acid tails, they won't pack as tightly and they'll be more fluid. So the fluidity of a membrane made up of phospholipids is determined by how many of the fatty acids are cis versus trans fats. Fatty acids, I mean. I keep going over here. I want to be over here. This is what I was doing. If you were watching the video, I was waving my hand. Cool? Very exciting, very exciting. Okay, so phospholipids, super important, all right? Steroids, cholesterol. Oh, we just talked about it. We just talked about cholesterol, all right? Cholesterol is a steroid. What's a steroid? it is essentially a lipid with a bunch of ring structures in it. So remember last week when I showed you the ring structures? If you look at this, you can see, oh, this is all carbon and hydrogen with one little hydroxyl group coming off of here, right? Look over here, cortisol, that's a steroid hormone, all right? It's like a stress hormone, okay? It's got its little carbonyl group over here, all right? But instead of being a long chain, it's linked up into rings. But it's all hydrogens and carbons, so it's still going to be a nonpolar molecule. So it's still going to follow our rules of being hydrophobic, like dissolves like, all that stuff. All right. We will talk about steroids again when we get into cell-to-cell communication because, you know, steroid hormones are super important. They are. They are. They have a special function due to their ability to be soluble in nonpolar solutions. It lets them cross membranes. I'll just tell you. I'll give you the spoiler, okay? But that's enough about it for now, I think. Delicious. Proteins. We've talked about the lipids. Let's get into the proteins. We got to get into this topic. We got 10 minutes. That's okay. That's okay. Proteins. You consume them. You are made of them. If I was to be entirely desiccated and then ground up into a powder and then analyzed, about 55% of what was in that powder would be protein. All right? Proteins are super important. They serve a lot of different roles. All right? The protein building block is the amino acid. And the amino acids are joined in a true monomer-polymer sort of framework, right? Where it's a long chain of these different amino acids, and the amino acids, their variability and what order they're joined in will determine the properties of the polymer, okay? Cool. This is the central dogma right here. It states that DNA is read and used to direct RNA, which is then read and used to direct the synthesis of a protein. This will be super important. We're going to talk about it more and more and more. But think about this, right? 55% of the dry weight of a cell is protein. You know your DNA is important, right? Like, we don't want bad things to happen to our DNA. We don't want mutations in our DNA. So why not? Because the DNA includes all the instructions for making the proteins that make up you. And we'll talk about the different types of proteins and how they determine the complexity of the organism, but the instructions are all encoded in the DNA. Solid? I love it. Thank you so much. Protein types. Let's talk about them. There's a lot more types of proteins. So far, it's been like, oh yeah, there's structural ones and there's energy storage ones. And that's been kind of everything. I'm like, that's structural carbohydrates, and there's carbohydrates for energy storage. And then for the lipids, they can be for energy storage. They can be for insulation, right? They make membranes, they have a bunch of different functions. Proteins have more varied functions, okay? Let's talk about them right now. Enzymes. Enzymes are proteins. Enzymes are biological catalysts. They encourage certain reactions to happen. So we already talked about lactose being a sugar and lactase being the enzyme. An enzyme helps something break down, or it helps a reaction happen. We'll get into way more detail about it. I'm just trying to keep it simple for now. So check it out. Enzymatic proteins, they help to break things down. They help reactions happen. Defensive proteins, all right? What are antibodies? Antibodies are proteins. They're proteins that your body makes that can target and stick to invaders, and then your body recognizes the antibody and says, that's a bad thing, let's get it. All right? Proteins, defensive proteins, storage proteins, ovalbumin, all right? So proteins aren't usually really used for energy storage, all right? You know that if you've ever Okay. Your body, right? You don't eat enough food. The first thing your body does is it targets the fats, all right? And it targets the sugars, then it targets the fats, then it starts to eat on the proteins. So there's some energy in protein, but usually you want to avoid letting your body do that. So you try to eat a bunch of protein in your diet if you're not eating a bunch of sugar, blah, 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 all these things, and then maybe eventually you'll be happy. That's what I just tell myself as I eat the 8th egg of the day, So here we go. If proteins are not used for energy storage, then what is a storage protein for? A storage protein stores amino acids. So you need amino acids, the building blocks, to make up all these proteins. Think about the chicken that's going to develop inside of this egg. It needs a bunch of amino acids to build the chicken, okay? So a storage protein stores amino acids. It can be broken down, releases those amino acids, which can then be used as the building blocks for the proteins. All right. Moving forward. Transport proteins. Transport proteins transport things. Look at this phospholipid bilayer right here, this cell membrane. All right? It's got the little phospholipid diagrams that I told you about. Okay? So your cell membrane, is made up of phospholipids, where the hydrophobic regions all associate. That means that this is a hydrophobic solution. That means that the molecules that dissolve in the water outside of a cell, the aqueous solution outside of a cell, and the aqueous solution inside of a cell, they won't freely cross the cell membrane because they are hydrophilic molecules. Reasonable so far? So you use special proteins that are embedded in the membrane to help transport things. These are transport proteins. Sometimes they use energy and they actually pump things, and sometimes they're just channel proteins, and all they do is make a little hole in the cell membrane. Cool? Question? Ah, nice. Good call. Okay. Proteins as hormones, they move through your bloodstream, they trigger responses. Insulin, okay, that's a protein. Thanks, it's way nicer closing the door before the noise comes in. Receptor proteins, all right? So if I'm a cell and I need to receive a molecule, receive a signal, okay, that signal can't get into me as a cell unless it's hydrophobic, right? The whole thing about the barrier. So you can have these receptor proteins on your surface that can receive a signal, okay? They detect something outside of the cell, and then inside the cell they go like, hey, go do something, break down some glycogen, all right? It's time to break down glycogen. So like for these, hormonal proteins to function, as an example, you would need receptors that can detect the hormone. All right, so receptor proteins. You're going to see a lot of this about these proteins get embedded in the phospholipid bilayer, in the cell membrane. Additional types of proteins, well, you got structural, boring, we already know what this is. So one example would be like collagen, okay, connective tissue inside of your ears and things like that, Structural proteins, they make like gooey, they're not like hard like a cellulose. Anyway. Then you've got these contractile and motor proteins which make your muscle tissue up. All right, motor proteins, they essentially just, here we go. So we've got like a fiber, okay, a long protein fiber, and you attach a motor protein to it, okay? Well, it didn't quite work. You attach a motor protein to it, all right? Then you blast energy into it, and it makes it move and go, right? Like this. And so a bunch of those working together contract your muscles. They do that, all right? So contractile and motor proteins, they work on larger scales to make your muscles flex, They work on smaller scales to move little things on the cell level. We're going to talk when we get into our cell bio about motor proteins and how they move around vesicles within the cell and how they lead to a phenomenon known as cytoplasmic streaming. All right? Which, it's not on Twitch, okay, guys? I'm talking about cytoplasmic streaming. Wow, we have so much fun. I'm so in touch. You know? I'm hip. Amino acids. All right? Let's talk about them. We got another couple minutes. I like to use all of the time. All right? You know this about me. You hate this about me. But you're stuck with me. All right. Amino acids. Here they are. The amino acids are the monomers that are joined into a big polymer. I'll do the hand thing again. Okay? And they differ in these side chains that project off of each amino acid, right? I'm not going to do the thing I almost did where I flipped you all off. We're not going to do that. It's hard to avoid, though. This is really difficult. My hands can't do this. Okay, here's the thing. You join a bunch of these into a sequence, all right? And they have all of these different side chains projecting off of them. So look, this purple portion of all the different types of amino acids, it's the same, okay? That portion is just repeating in this thing we call like a backbone structure. But projecting off of each amino acid are different, well, they're called R groups here, which is a general term meaning that they differ, okay? So we have these side chains, all right? And those side chains have different characteristics, like being nonpolar, being polar, or being charged. And what that does is when these are joined into a big long sequence, it makes a protein that has regions that are nonpolar, regions that are polar, and little pockets of charge. All right? And that's going to make this big structure that has distinct regions and a distinct shape and a distinct form and a distinct function. Okay? Here's an example of the peptide bond forming between amino acids. This is why a protein's called a polypeptide, because there's a bunch of peptide bonds in it. We'll talk about this and proteins on Wednesday, which will feed perfectly into your lab on Thursday. We are going to be extracting and quantifying proteins. Thank you all for being a part of Biology 105. I will see you on Wednesday. Have a wonderful day. Have a wonderful day. Thank you.